Every 19 minutes, an Australian suffers a stroke. It is one of our biggest killers. And now, a radical treatment for stroke victims using human amniotic fluid from pregnancy is being developed right here in Melbourne. And I'm pleased to say Professor Chris Sobey from the Trobe University joins me now in the studio to discuss this. Professor, this is a really exciting project, no doubt. How long have you been working on this?
Yeah, thanks, Alicia. Yeah, for about 12 years, together with our collaborators at Hudson Institute and Monash Health, we've been studying these cells as a possible therapy, especially thinking about the people, the 80% of stroke victims that can't get clot targeting therapy fast enough.
So why amniotic fluid? What is it about amniotic fluid that has these sort of properties, these healing properties, I guess?
So it's actually the cells from the inside of the sac, so the fluid's inside the sac and the cells line the layer. They're called epithelial cells. Our collaborators at Hudson have pioneered a method to isolate and prepare these cells. They can be frozen down and then thawed quickly. They've got amazing properties. They can be injected intravenously. They find their way to where the injury is, so they follow the signals sent out by injured tissue. They're very safe. They don't form tumours, and they don't require immunosuppressant drugs. So from one donor to a recipient, they can be given quite well.
It's just amazing, isn't it, when you think about something that really is from the start of life, is potentially helping people that otherwise would be at the end of their life. Let's talk about the trials that you've done so far. So as you say, you inject these cells, and 8 stroke victims were part of that trial. What happened?
Yes, so in the pipeline towards developing a therapy, we first, of course, have to verify that a new therapy like this is safe. And so we've infused these cells into 8 different patients that have had a stroke within the past 24 hours. And fortunately, there were no problems observed. And so this is a safety trial. Some of the patients did very well, and that's encouraging, but we're not in a position to know whether it was the cells that accelerated that recovery or not. But it's given us encouragement and certainly enabled us to now think about a larger phase 2 trial where we can look at an effect of the cells.
And the hope is, of course, that there's the potential that it could reverse some of the damage.
Reverse or prevent. We think the cells really quieten down the inflammation in an injury like this, and the inflammation seems to spread and expand the injury and make it worse. And so if they're given early enough, potentially it could stop that getting bigger. But even later periods of administration, we believe they exert sort of pro-healing properties.
Amazing. So stage 2 trials are next, and today you're celebrating a very welcome donation. Yeah,
it's an amazing donation from the Beluga Foundation, $6 million that really gets us a long way down the track and gives us the ability to now plan this phase 2 trial. So there'll be 78 patients, 3 groups, a placebo group, a high dose and low dose cell group, multi-centre trial around Australia. Yeah.
You might not want to do this or crystal ball, but what's the potential of this, do you hope, or do you think?
Yeah. Look, best case scenario, this becomes a routine therapy that's in the freezer of every hospital and clinic, and if somebody has a stroke, the cells can be thawed and infused, a little bit like albumin is as required in clinics.
And when can we expect, I know it might be hard to say a timeline, but when could you expect this to be widely available if all goes to plan?
Yes, that is a few more years away. It'll be a couple of years before we get the results of these phase 2 trials, and that will give us a real idea of the size of the effect. Then we would need further phase 3 international trials, but perhaps 5 years or so we would be getting close.
All right, Professor Chris Sobey, congratulations on all of your work so far. Of course, as you say, it's been more than 10 years of